Hi, I'm George Hammond, conference producer at Technology Networks, and I'm here to introduce our next talk, the use of spatial technologies in the Human Heart Cell Atlas. I'm incredibly pleased to welcome Dr. Anna Wilbrey-Clark and James Cranley. Anna is a senior staff scientist in Sarah Teichmann's lab, where she has worked on the Human Cell Atlas project for the past 6 years. Anna is focused on spatial genomics and transcriptomics technologies, specifically bringing in and scaling up new technologies to support Sarah Teichmann's research activities, whilst James is a Wellcome Trust-funded PhD student who joined the Teichmann Group in 2021. He received his medical degree from Oxford University before undertaking an NIHR academic clinical fellowship based in Cambridge. A very warm welcome to you, Anna and James. Following the presentation, we will also have a Q&A session, and we welcome any questions that you may have. Please ask questions using the Q&A system to the right-hand side of the video player. And now, without further ado, I will hand over to Anna and James to begin their talk.
Thank you very much, George. So as George said, I'm Anna Wilbrey-Clark. I'm from the Sanger Institute, which is in Cambridge, just in Hinxton, down south of Cambridge. And I work in the laboratory of Dr. Sarah Teichmann, which works on the Human Cell Atlas project. So what I'm going to talk to you about is a bit about what the Human Cell Atlas project is, and also about some of the spatial transcriptomics technologies that we use for this project, including some of the pros and cons of the different methods that we use. And then James is going to take over and talk about some of the data that's been generated, including using some of these spatial technologies for his Heart Cell Atlas work. So the Human Cell Atlas, our mission statement is to create a comprehensive reference map of the types and properties of all human cells, which are the fundamental unit of life, as a basis for understanding, diagnosing, monitoring, and treating health and disease. So basically what we are trying to do is to map all of the cell types in the human body, and we're doing that using RNA sequencing methods primarily. So we're defining cell types based on the genes that they are expressing. The image that I'm showing here shows a number of the tissues and organs that we are working on as part of this project. So you can see it's quite comprehensive. And we're also studying throughout fetal development and also throughout aging. So we're really trying to cover the whole lifespan of human development. Something that's starting to happen more recently in the consortium is that we're starting also to begin to look at models in a dish, for example, organoid models, and trying to understand those and improve models using the data that we're generating. So this consortium, it is a global consortium. It actually started back in 2017. And as you can see, the consortium has expanded a lot over time. So to date, we've now got 95 different countries with over 3,000 members involved in this. And all of us have the goal of trying to map all the cell types in the human body. The consortium is co-chaired by Sarah Teichmann, who is at the Sanger Institute, and Aviv Rajiv, who is at Genentech in the USA. And really, we aim to be as comprehensive as we can in terms of looking in detail at cell types, so even trying to identify small cell populations. In terms of covering the lifespan and development, also we're trying now to start to collect more ethnically diverse samples and to try and produce a comprehensive atlas. We've focused primarily on the healthy human state, so trying to generate that baseline reference map of what cell types are in a healthy human individual. The consortium is very multidisciplinary, so we work extensively with clinicians, pathologists, and many different scientists in different fields from around the world. And all of the data generated should be open access so that anyone who knows how to analyze this data can access it and use it. And as you can see from this image, this is a very international project as well. So in terms of why we want to run the Human Cell Atlas project, there are many potential uses of this data. So in terms of thinking about regenerative biology and thinking about those models in a dish, if we understand the development of humans and understand the cell types, how they develop and what molecules are involved in causing that development, that can help us to improve the models that we have in a dish that you can then treat with different therapeutics to see how that affects the cell types. Also, in terms of looking at disease mechanisms, so I mentioned that we are really trying to study the healthy human as a starting point. However, this project's been going for 6 years now, and we do have some quite good atlases of certain organs. For example, for the lung, there's now quite a detailed single cell atlas. So we are now more and more starting to study disease states and compare disease conditions against healthy conditions. That's also obviously potentially useful for drug discovery. And it's useful for looking at drug toxicities as well. So if you're a pharmaceutical company and you are developing a small molecule, again, that will typically target a protein. If we understand from the Atlas data where every gene is expressed in every cell type in the human body, then by default, we should understand where every protein is expressed in every cell type in the human body. So if you're developing a small molecule that's targeted against a particular protein, then you should be able to say your molecule is directed against a liver protein involved in liver disease, then you should be able to look at our data and say, oh, well, that protein actually is also expressed in the heart. So we have an idea to think in advance that we might want to look out for side effects on the heart. And it helps to think about those things in advance. Also for diagnostics and personalized medicine. And basically, there are a lot of potential uses from this kind of data. So these are the tools that we use. The Atlas is primarily involved in single-cell and nuclei RNA sequencing on dissociated human tissues and on spatially resolved methods, which is what I'm going to talk about. And there's also a huge data analysis and storage piece and making that data openly accessible through the data coordination platform. So I'm going to focus just on the spatial methods that we're using in our laboratory and the pros and cons of those methods. So I'll talk about RNA-scope, in situ sequencing, which has become Xenium, the different flavors of Visium, and also Curia. So imaging-based, to start with RNA-scope. So RNA-scope is an in situ hybridization method where you can have fresh or FFP tissue sections. You treat them with these double Z-shaped probes. So you have to have two of these Z-shaped probes bind next to each other, which gives you good specificity. You'd also get in your vial around about 20 of these double Z probes for each RNA target. And then once these are bound, then you apply this hybridization tree, which is heavily labeled with a large number of fluorophores. So that gives you a good signal to noise ratio. And the ultimate readout will be that you'll get one spot of fluorescence indicating one RNA molecule in the tissue. So you get lots of spots to demonstrate presence of RNA. So these are the kind of readouts that you get. For us, there are a few RNA-scope methods, and we've been using the 3 to 4-plex fluorescent method mostly. This works really well for us when we have generated a large single cell data set. We've identified some new cell types that we're interested in, and we want to validate, firstly, that those cell types really do exist, rather than just being clusters on a plot, and we want to see where physically they're expressed in space. And we found this assay to be very robust and easy to run and excellent for doing that kind of work. You can also automate it on a Lycobond or Ventana system. And if you find that you are having issues, because one of the issues you can get with the fluorescent method is that some tissues have high autofluorescence in certain channels, especially the green. And if you're really having problems, there is a chromogenic readout that's 2-plex as well. So in terms of the issues, the challenges with this method, as I mentioned, autofluorescence can be a challenge. Also, if you want to quantify and the spots and segment the cells, that's also challenging. But what we found is for our purpose, we're trying to demonstrate existence of particular cell populations, and we haven't actually needed to do that kind of spot quantitation for this method, really. We found that it's sufficient without doing that. There is also a high-plex method for doing RNA-scope, and that looks at 12 genes. However, we found that to be quite manual and time-consuming, and actually it has lower signal than the 3 to 4-plex method because you don't do as much amplification. It's a cyclic imaging method, so you don't do de-amplification. And so we have not found that to be as useful for us because of the signal-to-noise ratio not being as good. So the second method I want to speak about is in situ sequencing. There are a few iterations of in situ sequencing. The most recent is this hybris, where you use padlock probes, which you can see here, that bind to your mRNA. The padlock probe has 2 arms that bind to the RNA with a little nick in between them. And so you ligate the nick together, and that essentially generates a circular molecule so that you can then amplify around that circular molecule, and you can just keep amplifying and amplifying using a process called rolling circle amplification. And that generates a large blob of DNA, basically, that deposits on your tissue by just continuously amplifying around this circle. Within the padlock probe, you will have a barcode that is specific to your particular transcript that you're interested in. And you can have 100 of these different probes. So the way that you then read out which transcript is which is similar to doing a sequencing experiment in a tissue. So you detect the probe sequence, and then you read out each of the base pairs in that probe using fluorescent nucleotides. And you do that through rounds of cyclic imaging. So you do 6 rounds of cyclic imaging. So you'll get a readout that looks like this. So there are 6 base pairs in the barcode sequence. Say in your first imaging cycle, you had a G as your first base pair, and that's lit up with the green fluorophore. So you'll see that spot of RNA will be green in the first cycle. You then strip it, and then you'll probe again with the new detection probe and say that your next base pair is a T, then that will light up, say, blue, and then you read that out as well. So ultimately, you'll get a barcode sequence generated by watching that one spot of RNA and seeing how it changes color throughout the cycles. You can read out what sequence of that barcode is, and that tells you what gene that is. So the advantage of this method is that you're now looking at more like 100 genes using this method. However, it's a very lengthy and manual process. It takes 9 days if you run this manually, and you have to do these cyclic imaging rounds. And the thing that we've really found is that the image registration is very challenging. So that means that if you look at the image down in the bottom left here, where you can just see 2 cells with a number of spots of RNA in there, you have to be able to monitor every single one of those and line up the images so that every spot is lined up exactly, and you can see the color of the spot change to read out the barcode. So that's quite difficult to do. However, this has now been to some extent replaced with Xenium. So I'll speak about that in another slide. Before I move on to Xenium, I just wanted to show one image of the in situ sequencing data, as I don't believe that James will have in situ sequencing in his presentation. So this is a 90 gene in situ sequencing panel, which was generated for the human fetal limb. And the 90 genes have been divided into broad cell type clusters here. So you can zoom right down to the single cell level in this, but this is the overview of the image. So you can see the epithelial cells in green around the edge of the tissue. In blue, you can see the chondrocytes, which mark nicely the developing digits in the limb. And you can also see endothelial cells and other cell types. Quite nicely in between these digits were detected a number of macrophage populations, which is interesting because they're thought to be involved in digesting away the webbing. At this stage of development, the limb is webbed, whereas when the baby is born, obviously you have digits, not webbed feet. So they identified a number of interesting things from this data. To move on to Xenium. So Xenium, you can increase the plex again now to more like around 400 genes. It's based on in situ sequencing, but they've made some modifications to the protocol, including smaller spot size. Generally, it's best to use low expressed genes. So I did do an in situ sequencing experiment with a higher expressed gene, and it can obliterate the signal from your low expressed gene. So really the most important thing is to focus on the panel design when you're doing these experiments. Xenium has a very easy workflow. It does the cyclic imaging on the machine, and it also does that spot registration. So it lines up the images for you, calls which transcript each spot is, and does the very basic cell segmentation. The imaging area is quite large, and 10x also have panels pre-designed that you can start off with if you want to. You can also do H&E staining once you've finished the Xenium run, and also immunofluorescence staining. So it has quite a lot of utility, but on the negative side, Xenium is expensive. So especially if you want to run a custom panel, so you want to add some of your own genes, then you need to look at running about 16 slides, and then that becomes quite expensive to do. So in terms of sequencing-based methods, really the primary method is Visium. So Visium started off back in 2016 out of the Lundeberg lab with 100 micron spot size. That was then purchased by 10x and became Visium in 2019. They made a number of modifications, including reducing the spot size to 55 microns. That's still not single cell resolution. We are hoping that this is going to become Visium HD soon, and that the resolution is going to go down again to closer to single cell resolution and more like 8 micron bins, but that's not quite there yet. So at the moment, we're working with the 55 micron resolution. And there are a few different flavors of Visium, which can be a bit confusing. So I thought I would go through the different methods to compare them. So the original method was the fresh frozen protocol, which is a manual protocol. And you're capturing your transcripts with poly A. So you have your glass slide, which has a grid of spots arrayed on it, 55 micron spots. On each of those spots, you have poly T to capture your poly A mRNA. You have your UMI, and you'll have a spatial barcode. So every spot has a different spatial barcode. You then generate a standard 10x sequencing library from that. So you take your tissue, you lay it on, and then you release the RNA, bind it to the spots on the slide, and then you generate a sequencing library. And because every transcript has a spatial barcode, you can refer which spot it came from, and therefore how the gene expression is different across the tissue. So this method has been working quite well for us. It generates quite long lengths of cDNA. I've put full length cDNA, although you do chop up the cDNA to just detect the 3' ends when you do the short read sequencing library. The alternative to that is 10x now have a machine called the cytosist, which runs a probe-based protocol. So the advantage of this is that it was designed to work with FFPE tissue, in which your RNA is very fragmented, and therefore you can't do a normal reverse transcription-based protocol. In this method, you have probes that target 18,000 genes in the genome. And for every RNA you're interested in, you'll have a left-hand side and a right-hand side probe, which bind next to each other on the RNA. They're then ligated together. And then you can wash away the unbound probes, release the probes onto the gene expression slide. And because these actually have a poly A on the probe, they can then bind to these Visium slides. So they'll bind to the spots. And then again, you can then amplify from the poly T capture to add the UMI and the spatial barcode so that you can identify where within your tissue section each transcript came from, which spot it came from. So, and you get a quite small size library because of the probe size. Another method that is similar in principle is Curio. So the idea in that you are capturing with poly T, so you're capturing your poly A mRNA and you get your barcodes. That's similar to Visium. You have your UMI and your barcodes. That's similar to Visium. But the difference is that Visium uses a glass slide where your oligo T is printed in spots in a grid onto the array. Whereas for Curio, you're using 10 micron sized beads that have the oligo T on it. So that obviously gives you a big advantage in terms of resolution. You're now at 10 micron resolution, which is closer to single cell. The tiles are pre-imaged by Curio, so they know exactly which spot is in which position, and that information is provided in data analysis. And they have 2 different tile sizes. Similar to Visium, you still get quite long cDNA lengths, so you'll get around about 1,000 base pairs cDNA length, but we have found that the yield of cDNA is very low from Curio, and then you do a next era library preparation. So I thought it would be useful to compare the Visium and the Curio and the pros and cons of these. Both of these methods, you have 2 different tile sizes, 2 different capture area sizes for different size tissues. Visium, on the plus side, you do a hematoxin eosin stain or a fluorescent stain on exactly the same section that you do your gene expression experiment on. So you can overlay those directly. You also get this very long cDNA length for the fresh frozen protocol. It's whole transcriptome. You don't have to choose what genes you want to look at in advance. And Visium is FFPE compatible if you choose the probe-based method. Then you obviously don't get the long cDNA lengths, but at least you can work with FFPE tissue if you want to. On the negative side for Visium, at the moment, the spot size is 55 microns, which is definitely not single cell resolution. And you have to use fresh frozen tissue, for which you have to do a tissue, for the fresh frozen protocol, you have to do a tissue optimization. And that involves figuring out what time you need to permeabilize the tissue for the RNA to bind to the slide. Basically, that's just a very annoying step to have to do, and nobody really likes having to do it. For Curio, on the plus side is the resolution, really. It's a 10 micron size beads, so you get close to single cell resolution. You do get long cDNA lengths. Again, for both these methods, you still chop it up to do a short read sequencing library. It's whole transcriptome. Curio advertised this as requiring no tissue optimization. Having run 3 different tissues now through the Curio protocol, we've seen one worked really well without any optimization. One was still struggling to get to work. The other one we had issues with to start with, but now seems to be working quite well. So I would argue that tissues definitely vary in how well they work in this protocol, and it may require some optimization. In terms of the cons for Curio, so your H&E is not going to be on the exact same section as you get the gene expression. It has to be a consecutive section because the tile is white, so you can't image through it. It's not compatible with FFPE, and it does not cope well with low expressed genes. So even if you have an abundant population, you'll tend to lose those low expressed genes that you may pick up in the Visium. Also, the library is quite low complexity, so you need 10% Phi-X in your sequencing run. And for the bigger tiles, the 10mm ones, you need 2 billion reads per tile for sequencing. So it's quite expensive on sequencing. So just to summarize what I've talked about, so I've spoken about the human cell atlas and our goal of mapping all the cell types in the human body. I've gone through some of the different single cell, the spatial genomics methods that we use, RNA-scope in situ sequencing, spatial transcriptomics, and Curio. And in terms of thinking about the future, I hope that the spatial field will move more into doing more multi-omic methods, also doing more multiplex protein staining, because that's something is starting to certainly happen, but we don't do a lot of it in our lab. I think we need to do more. I hope it will also evolve more into 3 dimensions as well. And for the Atlas project, I think we're going to be starting to do more and more work studying disease and models, cell models of disease in organoids, going more down that kind of route. So just to say thank you very much to everyone in the Teichmann group, all our support services, and especially to the tissue sources and the patients who donate samples to this project. And if anyone watching this is generating this kind of data, and you want to join us and contribute your data, please do come and register at humancellatlas.org/register. So I will just pass over now to James, who's going to talk about some of the heart cell atlas work. Thank you.
Hello there, my name is James Cranley. I'm a clinical cardiologist and also a final year PhD student in Sarah Teichmann's lab at the Sanger Institute. I'm going to present some work on our profiling of the human cardiac conduction system using a mixture of spatial transcriptomics and other multi-omics as part of a sort of showcase of some work that can be done with these technologies. And I would encourage those who's interested to read the original paper. This talk is going to focus solely on the conduction system, but there were other niches of the human heart that were explored in this using the same techniques. So what is the cardiac conduction system? Broadly, we can divide it up into the nodes, which are highlighted in red. The nodes contain spontaneously firing cells called pacemaker cells. So these cells will spontaneously depolarize, repolarize, and repeat. And then down in the ventricles, we have the ventricular conduction system, which is comprised of the AV bundle and bundle branches, and also the Purkinje fibers or Purkinje cells, which are the distal ramifications of that system. And the purposes of these systems are to generate the rhythm in the nodes. Normally, the sinoatrial node is the leading pacemaker in the heart. And then the blue system, the ventricular conduction system, is designed to rapidly disseminate that electrical impulse down to the huge number of ventricular cardiomyocytes in the ventricles. And without it, we wouldn't have such a narrow QRS complex. If we look at the R wave on the ECG, or the QRS complex indeed on the ECG on the right, we can thank the ventricular conduction system for that speed. So why did we target this conduction system for study? Well, one thing is that the disease, so from a clinical perspective, disease is common. Different parts of the conduction system can be affected and will cause different clinical syndromes. The treatments we have are currently essentially to implant artificial electronic pacemakers into the heart. And these are prone to issues, especially around the leads, can get fractured or infected. So an understanding of the cellular process going on might help us understand how we could slow the underlying degeneration of the conduction system and avoid needing to put these devices in the first place. And then I guess from a different perspective, you could look at other cardiac diseases. arrhythmias such as atrial fibrillation, and some people would characterize those as being akin to conduction system-like cells, pacemaker cells perhaps, in the wrong places in the heart. So if we understand what the differences are between the working cardiomyocytes and the pacemaker cardiomyocytes, or the cardiomyocytes conduction system, that will be beneficial. This was our approach to study this delicate set of tissues. So we had ethics to receive hearts which were had been consented for both clinical transplant and for research, but then for non-functional reasons could not be used for transplant. So that might include HLA type mismatch or donor size or donor recipient size mismatch. So every year across the whole UK, there are a very small number of occasions where that happens. And in that situation, that scenario, we were primed to receive the hearts. It was a tremendous logistical effort because you had to have a sort of 24/7 rota and be available for the handful of times this occurred. And then we would go out to where the heart was and perform a dissection within 4 hours of it leaving the body and freeze down the tissue. We would then have these OCT embedded samples, which are, I think, a very versatile format to store your samples, because you can then or what we did was we would use light, just traditional light microscopy, bright field microscopy, H&E imaging to confirm that the structures had been captured in the block, and we had an external expert confirm that. And then with the same exact block, you can then both apply technologies such as Visium, and also you can liberate nuclei with mechanical dissociation and perform Multiome, which is 10x's paired RNA in a TAC-seq assay. Visium, I should say, is 10x's spatial RNA-seq. This is an example of the dissection that we had to perform. So obviously it was really critical to get a high quality dissection of the structures of interest, and actually the nodes in particular have very little anatomical landmark. They're invisible macroscopically. You have to work it out by adjacent structures. So it was critical at the start of the project to work with Yen, who is based at Imperial College London and is a sort of expert in the anatomy of the conduction system, learn how to do this dissection. And it was a sort of a learning process where I would do the dissection on porcine hearts and then get feedback and improve my dissection until when the human hearts came in, we were in a good position to take the samples quickly. Now that we have the blocks in OCT, we use traditional H&E microscopy to confirm the structures had indeed been captured. So on the left and center, we have the sinoatrial and atrioventricular nodes, and on the right, we have the atrioventricular bundle. And these, the images were sent to our external expert for confirmation, and she annotated them. You see the yellow borders where the relevant structures were. And we can appreciate just on HE imaging the significant histological differences between the non-nodal, so the working myocardium in the sinoatrial node region. So that's essentially normal right atrial myocardium, with the nodal tissue where we have smaller cells which are buried in a dense extracellular matrix. And this compared it was very comparable with the original sketches in the original description of the atrioventricular node by Tawara over 100 years ago.
So using the RNA-seq component of the multiome that we generated from these different regions, we were able to make an atlas, so annotating the different cell types, and you can see the UMAP projection there of the different cell types. In this study, there were 680,000 cells and nuclei. The new regions in this study were the SAN and AVN. And the AVN region include both the pacemaker cells and the AV bundle. At the bottom, you can see different sort of punch-in UMAPs of the annotated cell types, these are the kind of the new cell types that we annotated. So we have the pacemaker cells in the SAN and the AV node, as well as a population of AV bundle cells and a population of Purkinje cells. And we had there were some known ion channels predominantly, which we could use as marker genes. So once you've collected your suspension data, the single nuke RNA-seq data, and you've gone to the trouble of doing this careful annotation, you then want to map these cells back into their spatial coordinates, of course. So this is the kind of the truly integrative component here. So we have the some of our Visium slides along the bottom, and we used a tool called cellular to map the reference signatures, which are obviously from the single nuke RNA-seq data, into spatial coordinates. And what was very nice was that we could see the dashed lines were the expert's annotation of where these structures were, because with these Visium images you also get an H&E image, and so based on that she could annotate the structures, and it was very nice to see that the cell types that we had defined were popping up in their expected locations. So this isn't spatial data, this is data from the single nuke RNA-seq, but we could see that the pacemaker cells of the SA node and AV node expressed a whole bunch of ion channels, not just the ones we'd used as marker genes, which give them the ability to spontaneously fire. And also they seem to possess a very broad repertoire of G-protein coupled receptors, as well as the downstream machinery. So they have the capability to act as signal transducers, not only to the well-known neuronal inputs that they receive, but also to a wide variety of neurohumoral stimuli. We also looked at using both the RNA-seq and ATAC-seq data. We could construct gene regulatory networks for these cell types. So this is an activator network in the pacemaker cells, and we could see that in the grey are the transcription factors, and the smaller nodes are the target genes, and some of the target genes are coloured if they are ion channels or GPCRs, which are known to be sort of important in pacemaker cell function, and the edges are coloured if that interaction was supported in the ATAC-seq data. So we found FOXP2, which is a transcription factor not associated with cardiac pacemaking, seems to be important in this network and governs a whole bunch of critical ion channels. And on the repressor side, we could see that TBX3, which is a well-known governor, sort of master regulator of the conduction system, was borne out in our human data, and suppressing many of the working cardiomyocyte programs. So now, going back to the spatial analysis, trying to go beyond just mapping cell types into their spatial coordinates, we wanted to then find the multicellular niches that make up the heart. So one way of doing this, a simple method, is to use the annotated structures that the expert has annotated, so the nodes, and look at the relative abundances of different cell types in those structures, and calculate an odds ratio to see which cell types are enriched in those structures. And we could see that in the synovial node we have the pacemaker cells, a series of fibroblasts, and a specific macrophage population. And in addition, we had a population of neural cells, which we re-annotated as glia. So these are glial cells which release a neurotrophic factor. And then in the AV node, we also interestingly found a very similar composition, including these glial cells, which was, again, totally unexpected. So what I just described, which was basically looking at enrichment of different cell types in manually annotated structures, is one way of leveraging the vast amount of expert knowledge out there. Another way we did that in this paper was to compare structures that were annotated manually with structures that unbiased computational methods could find in the data. So what I mean by that is that we could, once you've converted your Visium spot by gene matrix into a matrix of spot by cell type abundances, that's using cellular, you can then perform non-negative matrix factorization on that data set and look for patterns, so that is groupings of cell types which are commonly found together across multiple slides. So it gives you the power to look across multiple slides at once and find groups of cells. So these are essentially multicellular niches, one could say. And then one would like to then compare those NMF-derived factors with the manually annotated structures, and we did that using a metric of effect size, basically looking to see how similar the unbiased factors were to the manually annotated structures. And sometimes what we found is that one of the manually annotated structures would correspond to more than one unbiased factor. And that was quite exciting, as I'll go on to show you. And then once you have your factors or even your manually annotated structures, you can then, it's a really good platform to then go on and do cell-cell interaction analysis, because one of the problems with cell-cell interaction analysis usually is that if you throw a whole bunch of RNA-seq data at a package like CellPhoneDB, you will find thousands of potential interactions, but what you want to know is what is biologically plausible, i.e. which cell types are genuinely often found together, and those are the interactions that you think are probably more likely. And so defining these niches either through unbiased methods or manual annotation lets you do that. So here's what I mean. So on the left, we can see a Visium slide of the sinoatrial node, and you can see that the expert has annotated the node region in red. But when we perform the NMF non-negative matrix factorization analysis on the spot by cell type matrix for multiple slides of the assay node, we can pull out two factors which we named peripheral node and central node factors. And if you then plot the loadings of those factors in space, you can see how the central node really is bang in the middle of the expert annotated node region, and the peripheral node loadings are around the side. And we can see the cell type composition of those two factors. So we recognize some of the populations that we saw from the odds ratio analysis, but this gives us a more nuanced approach. And also this is based on multiple slides, so it makes it quite a robust analysis. And essentially we find that the core of the sinoatrial node in humans has the pacemaker cells, as well as an activated population of fibroblasts laying down lots of extracellular matrix. And in particular, as I said earlier, we found this glial population in the core. And then surrounding the core, you have other fibroblast populations and macrophages. So this is just then plotting this is now just showing the corresponding abundances of the cell types I just described. So these are the sort of core node populations plotted in space, just to sort of back up the point. And then this is what I mean by then this allows you to reduce your search space when you're doing cell-cell interaction analysis afterwards. So here we know now that the core of this assay node contains these four major cell types, these sinoatrial node pacemaker cells, NC2 glia, and then these two fibroblast species. And so then that really reduces the amount of searching you have to do in the cell-cell interaction analysis. And we also were interested because we, based on the gene expression signatures for these cell types, we could see some hints that there may be synapse-like genes being expressed, especially in the glial cells. So we created a custom, we added to the CellPhoneDB library of cell-cell interactions with some transsynaptic type interactions that we sort of mined from the CNS literature. And interestingly, when you do that, you can see that the pacemaker cells in these glia have a sort of synapse-like connection, or at least are inferred to have. And so this is the sort of general model that we then came up with of what's going on in the human nodes. So we have pacemaker cells, and we know from canonical literature that there must be neurons coming in. But then the kind of other discovery we've made is that there's this population of glia there as well as a bunch of fibroblasts, and we sort of annotate quite a few of the potential connections there. And so this is just showing that we were able to see neuronal populations in the spatial data as well. On the bottom right, this is, I think, really amazing sort of validation of this nodal niche model that we're proposing. You can see the glial cells are stained with PLP1, pacemaker cells are stained with HCN1, and then there's a DAPI stain as well. And you can see how the glial cells really envelop the pacemaker cells. They have a very close, intimate relationship, and it was amazing because the diagram in the center was made months before we actually did that confocal imaging, but it's sort of eerily similar. You can kind of see the processes coming from the glia touching the pacemaker cells. So here's a summary of the findings from this paper, this is just a précis of it. So from a biological perspective, this was the first description of the human cardiac conduction system at single cell resolution. We identified the profiles of pacemaker cells, AV bundle cells, and Purkinje cells, and we showed definitively that the human SA node is comprised of core and peripheral regions, and we showed the cellular composition of those, and we also showed that this population of NGF producing glia are found in the core of the node. From a technical perspective, I think some of the learning points for me, and which could be transferred to other projects, were that the fresh frozen OCT embedded samples were extremely useful. They were very flexible, enabling us to do both spatial and suspension data generation from the same exact blocks, which really makes such a neat project. You know that the RNA-seq and ATAC-seq came from exactly the same nuclei, and then the spatial data came from the same bit of tissue, so it's all very neat. Cell location is a really powerful tool for mapping these defined cell types in spatial coordinates. But I think to kind of take it to the next level, inferring multicellular niches is then what you want to do. Here we used NMF, but there are many other potential ways of tackling this problem. Other people would perform clustering on the Visium data, for instance, to find groups of Visium spots, which could also be viewed as niches. And I think another really nice point in this paper was we basically relied heavily on this expert annotation, and we were able to link it with the cutting-edge, unbiased methods, and that provided the greatest insights in this paper. So final couple of slides. So I would encourage you, if you want to explore the data here, go to heartcellatlas.org, and you'll see the up-to-date data set on there, which includes lots of spatial data, so you can browse it, as well as the single nuke RNA-seq data. And here are the acknowledgements. This is a big team project. I'm very grateful to many people at the Sanger and Imperial College, and of course my co-first author, Kazumasa Kanemaru.